We are Group 45, doing Figure 4, Supplementary Figures 5 and 6, and Supplementary Table 3.
Figure 4 compared adult SPF ash given high and low dosage of the AAV vectors. It used both untargeted and sgRNA1 guide RNAs, guided vectors, as control and experiment, respectively. They used immunofluorescence with antibody staining to measure the amount of OTC-producing hepatocytes in the liver of each respective dosage. Figure 4a is comparing the adult mutant mice given the untargeted vector against the mutated with the guide RNA in place. There was no sites of OTC-producing hepatocytes on either untargeted vectors given mice regardless of dosage amount as to be expected. Mice administered with the guide RNA showed many hepatocytes producing OTC and roughly the same amount of staining in high versus low dosages, possibly due to the time of tissue collection being shorter in the high dosage mice. The OTC-producing hepatocytes showed higher sparseness compared to newborn mice. Figure 4b shows the discrete nature of the OTC hepatocytes of adult mice in higher details. The staining of individual hepatocytes is shown in green and the OTC is shown in red. In both low and high dosages, the OTC is shown to be isolated in each cell that it was produced in, especially with higher magnification. Figure 4c showed percent survival of adult mutated mice treated with both an untargeted and guided vectors of the AAV system in low doses. Severe decrease in survival rate is seen past 3 weeks as high toxicity was suspected. Figure 4d showed percent survival of adult mutated mice treated with both an untargeted and guided vector of the system in high doses. Severe survival percentage decrease, especially compared to the low dose vector systems, was shown to the result of study termination after just 2 weeks. Figure 4e shows the change in urinary uric acid amount of both control and experimental adult mutant mice with varying vector dosages. Untreated mutated mice showed an expected change in acid amount. In both untargeted mice, there was minimal change as the amount was localized around 0. In low and high doses of vector with guide RNA, there was significant change in the amount of acid with a large difference between the low and high dosages. Figure 4f shows the amount of ammonia in the plasma of both control and experimental adult mutated mice with varying vector dosages. Untreated mutated mice showed minimal amounts compared to other treatments. Both untargeted and low and high doses showed relatively low amounts of plasma ammonia. Ammonia plasma amount in low-dose guide RNA vectors showed elevated amounts. Ammonia plasma concentration in high-dose guide RNA vectors showed very high amounts that were significantly larger than untreated mice.
Supplementary Figure 5a shows the liver histology of the mice, and it looks very similar regardless of the dosage, if there were any, or if it was gene-targeted or not. This data was included in supplementary because it did not support the theory that there was residual urogenesis damage. This figure was most likely included because of the shocking findings in Figure 4c and d. Although the liver histology was comparable among the tested mice, their protein expression differed as seen in supplementary Figure 5. The ALT and AST proteins are serum transaminases that can be measured to detect damage to the liver. When the liver is damaged, there are higher levels of both ALT and AST in the blood. There are slight observed increases in ALT and AST protein expression in the low-dose gene-targeted mice and in the high-dose gene-targeted mice. However, it was not statistically significant compared to the control groups, so this figure would not be great to support the liver damage theory in the paper. Supplementary Figure 6.
Supplementary Table 3 showed adults had more deletions. Cas9 can make a cascade complex with other Cas proteins to cut targeted mRNA. However, this figure suggests that the deletions were most likely not due to the cascade complex because the low dose in adults had less Cas9 expression than the neonatal mice. Instead, the authors think the deletions could be due to NHEJ non-homologous enjoining in adults that differ from newborn dividing liver cells. Supplementary Table 3. The size of deletions between neonatal and adult mice was measured to compare the DNA sequences of the targeted region OTC. The researchers observed the majority of the deletions in newborn mice were 0 to 40 base pairs with about 1% indel overlaps, while the majority of the adult deletions were 0 to 20 base pairs, but about 9% of them were over 100 base pairs. Adults also had more indel overlaps, about 6%. Perhaps suggesting epigenetic modifications of the genome during development. The sequencing was done to try to determine how the DNA changes during development. The deletion sizes vary. The next step was to figure out why the deletion sizes vary in hepatocytes,